DNA probe. This is a cloned DNA molecule labeled with a radioactive isotope such as phosphorus isotope or sulfur isotope, or non-isotopic label, that is biotin, which is used in molecular genetics to identify complementary DNA sequences by hybridizing to them. So they are labeled, and uh, they represent as bases, and then when they make uh, complementary pairing with the DNA basis, then we can easily identify the basis of the unknown DNA. Therefore, they are called DNA probe. Hybridization means, from the name, the process of joining two complementary strands of DNA or each of DNA and RNA to form a double-stranded molecule. This hybridization is possible because of there is a base pairing rule. Adenine, the base adenine always pair with thymine, and guanine always pair with cytosine in DNA. This is a DNA probe. This is a DNA probe which contains the known base pairs. Then when it will be inserted into this normal DNA, then it will make a base pairing with a DNA of our interest. Then after that, after making of the hybridization between the DNA and the DNA probe, you can easily understand the sequence around which the probe make hybridization is the sequence which you can know from the sequence of your DNA probe. Alleles, these are genes governing variations of the same characteristic or trait that occupy corresponding positions that we call loci on homologous chromosomes. This is also called alternative forms of a gene. So you can find the definition of allele in different books. Is such, for example, these alleles are known as a variant of a gene, alternative form of a gene. They are caused by differences in this variation, or these differences caused by differences in the sequence of the DNA at a specific position within a gene. For example, let's say H is for tallness of a height and small h is for shortness. This sequence of the gene may make that individual be tall, A, T, C, G, and the other sequence C, A, G, T may make an organism or an individual short. That is allele. One of the different forms of a gene or DNA sequence that can exist at a single locus, and an alternative form of a gene that occurs at the same locus on homologous chromosomes. What is locus? The location of a particular gene on a chromosome. The plural is loci. The locus is a physical site. It is a site, just the physical site on a chromosome, occupied by a particular gene or other identifiable DNA sequence characteristic. We call this locus.